Assalamualaikum and good afternoon to Dr. Sohaili and friends. We are from Group 4, DNAwesome, and these are our group members. Today we are going to present on the conventional and molecular cytogenetics technique in human and non-human sample. But first we will start with techniques conducted in human sample. So these are our table of contents. Next. We have chosen this article from Cesar et al. 2017, titled Clinical and Microscopic Findings of Two Prenatal Cases with Inversion Duplication Deletion of Short Arm Chromosome 8. Next, I will briefly explain on the background of the disease. Short arm chromosome 8 inverted duplication deletion is a rare disease that is characterized by a complex rearrangement of chromosome 8, and it is commonly reported in postnatal period. However, up till today, there were only 4 prenatal cases of this disease that have been reported. And the study also reported on NRG1 gene, which is necessary for heart development, was duplicated in both new cases, and GATA4, a transcription factor which is implicated in many cardiac defects, is associated with the deletions on chromosome. The clinical findings of this disease include congenital heart defects, dysmorphic feature, intellectual disability, and central nervous system abnormalities. Next. The picture illustrates the most dysmorphic feature of patients with inversion duplication and with the disease. Next. A limited number of studies have highlighted on the submicroscopic changes that occur in this syndrome. Thus, this study presented on 2 new prenatal cases of the disease and conducted cytogenetics analysis to reveal the complex rearrangement that occur in the chromosome 8. So the first case is 39-year-old woman. It is her second pregnancy. CVS was performed at 12th gestational week. VSD and fetal echocardiogram were observed at 18th week. Meanwhile, case 2 is also a 39-year-old woman, a third pregnancy. The amniocentesis performed at 16th week of gestation, considering parents' anxiety and advanced maternal age. VSD and fetal echocardiogram were also observed at 19th week. Next, Hanis and Maitasha will explain more on the technique.
Okay, so for the techniques, there are 3 techniques that have been applied for this study. So for conventional studies, this study used standard protocol, but what makes it different is that this study used G-bending techniques, and the samples that they use are from CVS from fetus 1, amniotic fluid from fetus 2, fibroblasts from both fetus. And these 3 has 3 cell cultures that obtained from each. And the last one is parental blood samples.
Okay, so 20 metaphase plaques were analyzed for each of the specimens. So there were a lot of metaphase plate that was analyzed for fetus 1, fetus 2, and of course the parental blood. So the observation is as followed. We can see from the conventional G-bending technique, there is an addition of material to the short arm of chromosome 8. Okay, so this is the short arm, and you can see the addition here. It is longer, and the same goes to fetus number 2. Okay, and then the parental peripheral blood lymphocyte metaphase were normal. So this indicate that the rearrangement that happens in this fetus are de novo.
Okay, so for the second technique is the fish technique. So this technique is the same protocol of fish, but what makes it different with other studies is that this technique use probes that are specific to the terminal region of the short arm of the chromosome QS, AS, and control probes indicate the long arm of the same chromosome. And in this study, at least 5 metaphases were analyzed for each case.
Okay, so from the conventional technique, the results show the elongation of the short arm of chromosome is. So what they need to do is to ascertain that result by using the FISH technique. So the FISH analyzes using the subtelomeric region probe. So they because the conventional technique showed that the rearrangement occur at the telomeric region, so they use a special probe that targets the telomeric region of for the short arm and long arm of chromosome 8. So it verified the findings and revealed that there is indeed deletion of the 8 short arm in both fetus. So you can see here, this is the normal metaphase fish image, which shows this fluorescence here, but on this fetus 1 it does not show that same signal, and it's the same for fetus number 2, it is not compared to the normal one.
Okay, so for the ACGH technique, this study also used the standard protocol, but what makes it different with other studies is that the DNA extract from the chorionic villus and amniotic fluid of fetus 1 and 2 respectively. And this technique also was performed using the Agilent Human Genome G3 SurePrint 8 times 60K ISCA oligonucleotide microarray, and the data were analyzed using Agilent cytogenomics software.
What kind of probe did they use for this to color the DNA extracted?
Well,
Hanis.
for the sample DNA, they use CY5, ULS CY5 for the sample, and for reference, they use ULS CY3.
Okay, so the array CGH also was used to validate the result of both conventional and fish findings. So from this image here, we can see for fetus number 1, there is a deletion that occur at the 8 p arm 23.1 region, which is sized at 7.5 MB, and duplication size 30.7 MB at 8 p arm 23.1 till 8 p arm 11.1 in fetus 11, in fetus 1. So between the two segments, here is a region of intact zone. So this region is not changed, it's not structurally rearranged. And the same can be observed in fetus 2, a deletion at the same region, but differently sized, 6.7 MB, and duplication of the same size and at the same region in fetus 2. And there is also the intact zone here in between the deletion site and the duplication site. Okay, so the duplicated segment is size approximately 30 MB in both cases, and it can be seen that it is inverted. So this figure shows the abnormalities and the related genes in our fetuses and the reported patient with this disease. Okay, so we can note that all the GATA4 gene is intact and the NRG1 gene is duplicated. So this correlates with the array CGH analysis that shows amplification of NRG1 locus, which is this region that is duplicated, and the deletion of this region and the intact region of GATA4, which is here.
For possible future study, we would like to suggest another method that the researchers can use to detect chromosomal rearrangements such as M-band. M-band is a multicolor banding analysis which enables the precise and more rapid assessment of different regions of a single chromosome using region-specific paints to increase the accuracy of karyotyping
and to aid in the identification of chromosomal changes, especially in complex cases with multiple chromosomal rearrangements. So this method, okay. For example, look at this figure. This is the normal chromosome 3 and this one is the normal chromosome 12. The analysis using specific probe sets for these chromosomes, the researchers could prove the additional material on derivative chromosome 12 resulted from an inverted duplication of the end part of long arm chromosome 3. See, the terminal portion of long arm chromosome 3 has become the short arm for chromosome 12. Okay. Therefore, the researchers could use M-band using region-specific probe for short arm of chromosome 8 to further validate those rearrangements involving inversion, duplication, deletion observed in the conventional karyotyping analysis. Okay, next.
Now, if we could circle back to this image, this is what Aina was saying, right? This
Yep.
region here could be colored and can show that which region is deleted from this, which based on the array CGH analysis, and which region are duplicated and inverted, which is this
Yes.
region here, presumably, and we can differentiate all that by the different color of bands.
Hmm. To conclude, if there are any more reported cases suspected with this disease, similar techniques such as conventional karyotyping, FISH, and array CGHs should be applied as well to fortify the hypothesis of the study, which is the overexpression of NRG1 gene could influence the observed discordance between left and right ventricles and ventricular septicular defects or VSD in the fetuses. It is encouraged that the researchers could include the suggested technique, which is M-band, in the future to further aid in the analysis of the study. Okay, that's all for question 1.
Okay. Okay, next is the conventional and molecular cytogenetics in non-human specimen. Next, the table of content. The first one is background, and then conventional cytogenetics, FISH, CGH, and the comparison. As for the background, oh no. Okay, so this is the article that we refer to with the title First Description of the Karyotype and Sex Chromosome in the Komodo Dragon. For the background, Komodo dragon is a well-known predator and the largest lizard in the world, commonly found in Australia, Africa, Asia, and many islands in the Pacific, including Indonesia. So the sex determination system of Komodo species is determined by ZW system, where female is represents ZW and male ZZ. However, it is extremely difficult to determine their sex by external phenotypic characteristics. Therefore, this study investigated the sex chromosome of Komodo dragon by applying methods of conventional and molecular cytogenetics. Next, Shafira.
So the first technique involved in this study we use is a conventional method. So the protocol for conventional cytogenetic, firstly, we use the metaphase chromosome spread, which were prepared from the whole blood cell culture following the protocol by Pokorna. So we take about 40 microliter of peripheral blood for culture for a week. And then we enrich with FBS, streptomycin solution, glutamine, phytohemoglutamine, and lipopolysaccharide. And then the next step, we prepare the chromosome following the standard procedure, including calcimide, hypertonic treatment, and fixation in ratio of 3:1, which is the methanol and acetic acid. And then we stain with the GIMSA. We prepare the GIMSA staining for the chromosome, and then we do the C banding. For the C bending, we etch the slide and then we soak in HCl. After HCl, we soak in alkaline solution, which is the barium hydroxide solution, and then we rinse back into the HCl. After that, we do the DAPI staining. For the DAPI staining, we incubate the slide in saline sodium citrate buffer, and then we rinse in distilled water. After that, we stain with DAPI. So for the observation, from the figure 1AC, we can see that the gene sustained karyotype for the figure A is male and figure 1C is female. Whereas for the figure B and D is the C-banded metaphase. B is for male, D is for female. For the figure 1AC, the karyotype is 2N, which is equal to 40 chromosomes, which consists of 8 pairs of macrochromosomes and 12 pairs of microchromosomes. And then the figure 1BD, this reveal the C bending is to reveal the heterochromatin bending, which in the pericentromeric position, which reveal the two largest pairs of macrochromosome and the only one of the acrocentric pair of macrochromosomes. So based on the figure 1C, we can see that basically the Z chromosome could not be distinguished by any C bending or by CGH. So by integrating the C banded chromosome into the karyotype, we can see the W chromosome, which falls within the range of chromosome 9 to 12. Generally, in some species, the W chromosome is distinctively larger than the other microchromosome. But this species, the W chromosome is not larger than several other microchromosomes. So the W chromosome falls within the W chromosome is indicated that the species is the female type.
Okay, as for fish technique, the technique involved in non-human specimen is very similar as described in human specimen, which involves sample preparation, fish protocol, hybridization, wash and counter stain, and also examine. Sample preparation, the metaphase spread is prepared from the whole blood cell culture. And then denaturation of the sample and fish probe. The probe used is telomeric, rRNA, and microsatellite. And then the sample site were treated and then fixed and dehydrated. The next one, the fish probe will be applied on the sample site and incubated to allow the hybridization of fish probe on the sample. Next is post-hybridization washes were performed in formaldehyde to remove the unhybridized probes and counter stain with DAPI. The last one is image analysis using fluorescent microscope equipped with digital camera and also DP Manager imaging software to impose the source for observation. The first one, fish with telomeric probe. So the figure shown there, the telomeric probe provided typical signals restricted to the terminal region only of all chromosomes. And then the next one, fish with rRNA genes and microsatellite probes. The figure shown in the right side is the probe for rDNA bound to the pericentromeric region of the largest chromosome pair. As you can see the arrow there, the two arrow there. So the next one is fish with microsatellite probes is performed to identify the accumulation of AAT, CCG, and GATA microsatellite repeat in the motif and the W chromosome of Komodo dragon. However, this study did not reveal any significant accumulation on any of the chromosome. Next is CGH.
Okay, so the The third technique involved in this study is comparative genomic hybridization, CGH. So the protocol for the CGH, firstly, we label the DNA using the nick translation kit. The male DNA was labeled with biotin DUTP, whereas for the female DNA was labeled with digoxygenin DUTP. After DNA labeling, we proceed with co-precipitation. For the co-precipitation, we need the male and female labeled genomic DNA to co-precipitate with salmon sperm and sodium acetate. Then we proceed the preparation before hybridization. So the dry pellets were suspend and we denatured, chill on ice. But at the same time, the metaphase slide were treated with RNase and pepsin, and then fixed with formaldehyde, and then dehydrated, denatured, and then dehydrated again. And next, we do the hybridization. The probe was applied to each slide, and then we incubate for 48 hours. So after the hybridization, the slides were washed in formaldehyde. And then we incubate with the blocking reagent, which is Roche. And then the blocking reagent, including the avidin FITC and anti-digoxygenin rhodamine Roche. And then the slides were subsequently washed in SSC Twin 20 and then dehydrated through an ethanol series and air dried. So after the hybridization, we proceed to the mounting slide with DAPI. Then after we mount the slide, we image the slide using the fluorescent microscope equipped with digital camera. So the observation based on the CGH method, we can see that the figure 2, we can see the picture from the CGH itself is more clearer, and we can see the color that show is a red, green, and yellow. So based on the figure 2, A is for male, B is for female. And then for the figure 2AB, the male genome is stained with green color, which is the FITC. And then the female genome will stain with red color, will show the red color. For the combination of the green and red color will express the yellow color, which is the combination of both sex. So as we can see from the CGH image, we can see the W chromosome is expressed only in the female image. So it is clearly visualized by CGH where only one chromosome hybridized predominantly with female specific sequences in figure 2B.
Okay, now we will compare and contrast of the conventional and molecular cytogenetic technique in both the human and non-human study. So basically, both paper includes the conventional, fish, and CGH. So now we will compare one by one according to the technique used. Next. Okay, the first one is the conventional cytogenetic. So basically, in terms of the sample, in human paper, we can see that it uses 4 different samples, which are chorionic villus, amniotic fluid, fibroblast, and blood sample. Whereas in the non-human, it uses only one sample, which is the peripheral blood. So in terms of the staining, both human and non-human uses GIMSA staining. However, it differs in the banding where the human paper uses a G banding, in which it will stain the chromosome arm, mostly the repetitive AT-rich DNA, whereas for the non-human, it will banding of the chromosome is the C banding, in which it will stain the centromere. So for the cultural medium, for the human, we use amniomax, and whereas for the non-human, we use DMAM. So basically, the cultural medium, it depends on the samples, type of sample used. Next. Okay, for the fluorescent in situ hybridization, for human and non-human paper, it differs on the types of probe. In which the human, it uses subtelomeric probes, whereas in non-human, the paper conducted two types of fish. In which the first fish, it uses the commercial telomeric probe, and the second fish technique, it uses rRNA gene probe. Next is the fluorochrome. For the human, it uses CY3 and CY5, and whereas for the non-human, it uses CY3 fluorochrome. So for the counter stain pipe for both human and non-human, it uses DAPI stain.
Okay, next is on the comparative genomic hybridization (CGH). So on both human and non-human, it differs in the type of the CGH used. In the human, the article says it use the microarray CGH, while for the non-human articles, they use normal metaphase CGH. And in terms of the test sample, in the human articles, the DNA is extracted after the cultivation of the coronate villus and amniotic fluid materials, while for the non-human, the test samples is the male and female genomic DNA of the Komodo dragon. And next, the fluorescent label. For the human paper articles, the reference, which is a normal chromosome, will be labeled with CY3 DUTP, which will give the red color. But for the test sample is CY5 DUTP, which will give the blue color. While for the non-human, for the male genomic DNA, it will be labeled with biotin DUTP and fluorescent with avidin FITC, which will give the green color. While for female genomic DNA, it will be labeled with dihydroxygenine DUTP and fluorescent with anti-dihydroxygenine rhodamine, which will give the red color. And it differs also in terms of analysis, which is the human articles uses Agilent cytogenomic software. And for the non-human, since it is normal metaphase, it uses the analysis is performed using a Olympus fluorescent microscope with a digital camera. And with that, I hope you guys understand. Thank you.